Welcome to one of the interviews with Vascular Discovery 2026. I'm Peter Henke, co-chair of Vascular Discovery 2026, and I have the great pleasure of having Dr. Zorina Galis with me today to answer some questions related to her outstanding talk that was given during the Hot Topics plenary session this morning. So with that, you covered a lot of kind of history with regards to your role in the NHLBI to kind of spark and promote research and initiatives within vascular disease. I have a question related to earlier you talked about the, I think it was Gentech AAA. Tell us a little about that, the NITIS, and also where it's at right now.
Thank you. So first of all, I want to say it was an incredible opportunity to be there because I think working at the NIH for the past 15 years, we have to do a lot of work, and several people came after my talk and they told me, we didn't even know what you are doing at NIH. Now we learn more. I want to use this as an opportunity to ask other people who are considering alternative careers to come join us. I worked in academia before and also work in the industry, and I can say that this was the most rewarding job I had so far because we are able to enable science in the research community, and we have many ways of doing this. And while I presented several activities that I have been related to and involved in, some of the timelines started before I joined the NHLBI, the Heart, Lung, and Blood Institute. So what I was trying to highlight is the the continuous or maybe even relentless type of activities that we have to put in place. And now we are seeing those topics emerge from the portfolio. And actually, this is how I started. I thought about how would I select hot topics, and I could have done a lot of things. I started with bibliometrics. I tried various things, but then it occurred to me that one perspective that maybe the audience is not familiar with is the bird eye view that we can have at the level of the NIH. And many of us are extremely dedicated to identifying areas in need of enabling, and we do a lot of things to do that. So coming back to the specific question about Gentech, that started 20 years ago. So with the recruiting of the first patients in the genetically triggered aneurysm, at that time, we didn't know much about this condition. And so the big challenge was to develop a resource where we had actually samples, imaging, information, clinical information about the patients who were affected by this condition, and to find out what are the connections with the cardiovascular disease. So that started, before I was at the NIH. I identified one of the previous employees at the NIH, Dr. Tolnai. And what was very amazing about that once we built, and the lesson we learned, that was we built that resource, we already have now a research community that has been gathering around that resource and using it to basically apply for investigator-initiated studies. And we soon enough started a scientific event that was hosted initially by us. And as I said, they used to happen in maybe in Baltimore, but it became a biennial event, and it became more and more interesting. When the grant ended, as this happens, right, we always take a lot of measures to make sure that the resources are not lost. So there's a process that we call planning for sunsetting of the funding. So we knew the funding is going to end because all grants have a finite time. And we were already collaborating with Marfan Association and other professional and patient groups. And they saw the incredible opportunity to actually take over this resource. And now it's actually part of the Marfan Foundation, a special division of the Marfan Foundation, and they are continuing with a scientific event. And as I said, we can also watch the progression of science from initially asking questions, well, what do we need to even collect from these patients? Or what's the benefit of a repository? What kind of questions could we ask, to where now it's used to make clinical decisions? We already see clinical trials being promoted and expansion of all the clinical implications.
Yeah, that's awesome. And I think
Yeah.
the power of the NIH and bringing people together and supporting it, launching it, so to speak. And
Yeah.
actually, one of my partners, aortic researcher, attended the conference. I think it was in Europe, in
Norway.
Norway. Yes, yes, and
Yes.
big group of folks
Yeah.
focused on that. So that's
Yeah.
awesome. Another thing you highlighted was and I thought that was funny. You asked the three main AI agents, I guess,
Yeah.
for hot topics, and it was really pretty accurate. So, where do you see, particularly from your vantage point, artificial intelligence and large language models going forward?
So we are encouraged very vigorously to use AI, and we do that for many different purposes. One of them is obviously to try to identify important patterns, especially as they come from very large data sets, or even to scan the literature. We also try to use AI to improve our processes and make them more efficient. As you know, there are a lot of questions when it comes to AI, and I was asked also to talk about what may be the implications of using AI for clinical trials or decisions. And there is a lot of information already. So FDA had a big effort actually by the previous commissioner, Dr. Rob Califf, that looked at what would be the potential risks in using AI for that information. When it comes to more sort of basic information, we think it's enormously useful, because it can summarize a lot of things that otherwise it will take weeks. I actually used AI to suggest slides for that talk, where I was asking what are the NIH recommendations for use of AI, before we actually had such official recommendations. And I can tell you that I spent about 2 weeks doing research by myself. And then I thought, okay, what if I would use AI to ask this question? And I was actually I don't even know how to characterize my impression. I was actually upset, because within 5 minutes, it put together all the information that it took me 2 weeks to go to the FDA website and all other places. And it even suggested some slides. Now, the benefit is that I had done the research already, so I could tell that I was telling the truth.
Yeah.
So one of the things that we're always having to make sure is that it's not just generating information so it has something to say to you, right? So as I said, when I got suggestions for this topic, my first thought was, well, maybe they already know what I'm working on. Maybe they're trying to please me. It's known that ChatGPT is sycophantic. But then they came up with a quite good suggestion, to where they said the new lens on resilience, the new tools that highlighted was spatial biology, and then the new targets in inflammation post cantos. It was a great lineup. So I thought, excellent. And of course, it also was perfect for what I could have talked about, because I've been involved with these initiatives.
Yeah. No, it's we're at the forefront or the beginning of this, as it's going to just continue to explode. And I think it will be good in the long term. It's just like you mentioned a
Yeah.
minute ago, you got to be able to fact check a little bit
Right. Right.
with that.
So the new models, you can actually ask in your prompt. So I took a lot of classes actually for AI, which is nice. And NIH offers a lot of classes for us. They might be open for the public, I don't know, about the prompt engineering. So if you properly engineer the prompt, you are more likely to get a great response. So you could say, I want you to show me your sources. So they will show you the sources that you can go check on the source. So that's when you do some sort of very wide, let's say, encompassing question research. Other than that, language processing and some brainstorming. They're pretty good for brainstorming because I'm thinking about doing this. What do you think? And you can create as you know, agentic AI. Now, one thing to highlight is that I showed at the end that there are now so-called highlighted topics at the NIH, and you will find that among them there will be requests for people to think about how to use AI or use AI for specific purposes, so people can go review those and decide what they could use the AI for.
That's
So,
awesome. Do
yeah.
you have to have special qualifications in AI to put that in a grant submission, for example?
I think that's more at the latitude of the peer
Reviews?
review, as you know. I mean, they can comment on the fact that you don't have the qualifications. Always, we tell people that go ahead and make sure that you have somebody who has some track record in that area to
Okay.
have as a collaborator.
Yeah.
Now, when it comes to what we used, I also gave the example to where we wanted to create a machine learning segmentation of tissues from 5 different organs, histology slides, right? And we submitted this to an AI platform, which is Kaggle, that's hosted by Google. And none of those people had any idea about biology or medicine. They were all AI experts, enthusiasts. I don't know. There are people who make a living out of winning these competitions.
Yeah.
So
Yeah.
it was an amazing, for me, show of how you can, depending on what your question is, you can take people from a different field that has, in a way, I think it's very good. They had no preconceived idea of what this should be like, right? And then they competed, and then we obviously gave accuracy prices, and they were able to leverage whatever they did, although they came with zero information. There is also a lot of cooperation that happens online, and this reminds me, I used to be in academia, as I said, Georgia Tech. I used to teach something that's called problem-based learning, meaning that the students didn't know anything specifically about the problem we're presenting, but they will do the research to answer that question. But that's sort of the wide world. When you come in front of the peer review system, they will probably comment, what do you know about this?
Right. Right.
Show me
Right.
that you know something, or show me that there is somebody who knows.
Yeah. No, awesome. And
Yeah.
again, kind of the future. Along that line, you spent a fair bit of time talking about the Hub map. And
Hub map, yeah.
Thank you. And my question is that two kind of, I think, probably simple questions. One is that being used in medical schools for education? And number two, maybe an example how it's been used for a disease process exploration or refinement of whatever disease it may
Right.
be. Let's say kidney disease.
Yeah. So I want to start by saying that the acronym, and some people like it, some don't. I'm to be blamed, maybe. But I thought we start with HU, which is human. So we always only look at human data, and then hub and map. So a human hub for maps of organs and
Yes.
tissues. So we have started again from a zero sort of baseline, because at the time we didn't even have a way of harmonizing information. As you know, every scientist has a special name for the cells they're interested in, or what may be the biomarkers and so forth. So it took an enormous amount of effort to bring people together to agree on specific way of defining cells, tissues, and so forth. So we've been doing this for now 9 years. We are almost about at the end of it. So is it used already in medical schools? Not in medical schools that I know of, because I think that requires a process that usually has to be put in place by the medical school. Is it used by students? Yes, we have online classes, and anybody can take a class on how to use this. We have a lot of students who actually took the class and developed projects around it. We also had a very good sort of adoption of the virtual reality approach. And we see that around the world, people are interested in putting them in museums and other ways of informing people. We have organized 24 hours around the globe and around the map where we demonstrate all this and we engage people. So it takes a while, as I tried to show. Some of the effort took about 20 years to bring to the clinical arena. This started, the first funding was 2018, so we are not so far out.
Right.
As I said, we had a lot of papers already being published, and now we'll have another big package of 80 papers where it has been been used to look at all kinds of conditions. The two that I had suggested were using to look at the effect of aging on the kidney functional tissue units, these units that basically encode the function of the kidney, and you have tens of thousands of them basically grouped together in order to perform the function for the entire body. So the question was, what happens with aging? And what we found, because we had these technologies now, we can show there are certain characteristics that change. Now, by learning what changes with age, you could potentially try to intervene later. We also had studies that looked, for instance, what is the effect of sun exposure on the skin. And we showed that with exposure to UV light, these cell characteristics, distances to vasculature change, so skin is affected. The same with disease and so forth. So what we are hoping to do is to get to the level of single cell information to where you get these signals. There are prodromal, means before disease happens. So when people come with a condition, the disease is already at the clinical level. It has manifested, widely in their body or the whole body. What we want to do is to be able to have very early signals in the tissue that will let us know that something is about to happen, or something already happened and we didn't know. The other thing that I want to say is that we, by design, wanted to build this hub map to be a map of the normal, or whatever is normal for age for humans, because there are a lot of other consortia that look at disease.
Right.
And so we have actually a specific consortium on kidney disease that's also funded by one of the institutes, the Kidney Institute at NIH. And now they are using and they are collaborating with HubMap to find basically the normal equivalent for what they have been collecting from people who already have clinical disease. So it's already, and we have papers that have been published and more to come, where they use our common reference to actually make suggestions about what happens in disease.
Gotcha.
Similar, we are working with the tumor network, where all they collected is cancers. So most people, the immediate reaction is, let's collect these tissues in diseased people. Our reaction was
baseline.
How do you know what,
What starting point.
what the starting point?
Yeah.
And of course, there are a lot of considerations. How can you really tell that these are normal tissues? And suffice to say, I was trained also in pathology, so I believe that if the pathologist, who usually takes decisions for you, like anytime, during an operation or cancer treatment, is the pathologist who says, this is normal, this is not normal. So we ask pathologists to actually look at the tissues and verify that they are normal. Now, whether the donors might have had some other conditions, that's hard to tell. But what we collected is normal tissues from those donors, almost 500 of them.
That's an awesome effort and resource going forward.
Yeah, hopefully. I'm hoping this
getting the word out about it.
I know it was hard to actually. So the tissues that were selected for HopMap were selected by peer review. I have presented this everywhere in the vascular community, hoping that we'll have people who will submit proposal. But at that time, as I said, the vascular community did not have the expertise or the interest to look at single cell spatial technologies. So now we are seeing that
interest.
emerging.
Yeah. Last topic quickly. Maybe explain the idea of resilience within what you were talking about.
Yes. So I believe that it's a very important topic because actually how Dr. Collins, who the former NIH
Mhm.
director, pointed out, we are called the National Institutes of Health. However, again, the normal and the health may have not been always front and center. So the question is, we know a lot about why some people get sick or die. But we don't know very much about why the rest of us may be able to resist those challenges and persist and have not only a long lifespan, but a health span.
Yeah.
We're talking about health span,
Mhm.
staying healthy. So, what are the determinants of health? How can you use what you learn about these people to figure out how you could now enhance the resilience of or the health of people who don't naturally have these mechanisms in place.
Yeah.
So it's a little bit harder to figure that one out. However, now with all the large repositories and data sets, it's possible. So to give you an example, you can take people who have a high blood pressure, and a lot of them will go on and develop cognitive impairment, dementia, and so forth. Yet you have people who have very high blood pressure, but they don't develop cognitive impairment or dementia. So, what was special about
Yeah.
those people? If we can figure out what was special about those people, hopefully we can apply that to try to help the other people. So there are many examples like that. So it was again, I think it was the time when my own personal experience with my colleagues, losing colleagues very early to cancer. We knew they had cancer, but we didn't know why we didn't have cancer. So
Right. Right.
that's the type of question that you would ask. We also now are interested in what may drive our health on various trajectory and disease. Some people get immunodegenerative diseases, maybe atherosclerosis or Alzheimer's. We talked about that. Some people have badly regenerative disease, cancer, and so forth. So why some people get cancer, some people get cardiovascular disease, and one of the topics was in a way approached today by Dr. Libby also, who showed that the chip phenomenon.
Mhm.
It depends. It can push you this way and the other way, and that's how it was actually discovered, looking at people who did not die of leukemia, but they died of
Hearts.
cardiovascular,
Yeah.
right?
Yeah.
So that's,
Observation.
I think there is a lot to be learned, and I think that ideas can come from anywhere, including from my high school reunion. Yeah.
Well, thanks so much, Dr. Gallus. A fantastic lecture. It's been great having you at Vascular Discovery, and really appreciate you taking the time and effort to get here.
Well, thank you so much for the opportunity. I really wanted to highlight what the NIH can do for the community, because sometimes we either don't appear on papers or don't get to speak, but we actually are working
Yeah. No,
relentlessly behind the scenes.
much
So,
appreciated.
yeah,
Thank you.
thank you so much.